Hello and welcome back to the second of a series of video lessons in pharmacology. Each of these videos is foundational for successive videos, so I strongly suggest that you watch the first lesson before watching this one. In the first video, we saw how 5 common chemical functional groups may be bonded together to create various drugs. In particular, we discussed how hydroxy, amino, methyl, ethyl, and phenyl groups could be combined to create various drugs related to the compound phenethylamine. In this video, we will talk about a few more chemical compounds and introduce one more chemical functional group, the carboxyl group. The carboxyl group consists of a carbon atom bonded to a hydroxy group and double bonded to an oxygen atom. The carboxyl group is often written as COOH. Drugs that contain a carboxyl group are often referred to as carboxylic acids. Now recall from the previous video that we can combine a phenyl group, an ethyl group, and an amino group to form the compound phenethylamine. So all this begs the question, what do you get if you combine phenethylamine and a carboxyl group? Well, let's find out. Look at the alpha carbon of phenethylamine, the one that is bonded to the nitrogen atom. If we remove a hydrogen atom from this alpha carbon and substitute a carboxyl group, we have a compound known as phenylalanine. The first thing to notice about phenylalanine is that it contains an amino group and an acid group. This compound is in fact a dietary essential amino acid. This means that humans cannot synthesize phenylalanine de novo, so our diets must contain phenylalanine or phenylalanine containing proteins. Phenylalanine is one of the 20 amino acids that are used by cells to synthesize proteins. The name phenylalanine indicates that this compound contains a phenyl group bonded to the amino acid alanine. Again, notice the logic to this nomenclature. This lesson is about catecholamines, not amino acids, so we'll move on. Observe that the phenyl group contains 6 carbon atoms. The carbon bonded to the ethyl group is the number 1 carbon atom, while the carbon atom that is opposite this, indicated in green, is the number 4 carbon. The number 4 position is also referred to as the para position. This particular para carbon is bonded to 2 other carbons and to a hydrogen atom. If we remove the hydrogen atom and substitute a hydroxy group, we create a compound that I like to call hydroxyphenylalanine. However, everybody else calls this compound tyrosine. Tyrosine is also one of the 20 amino acids essential for protein synthesis and was first discovered in the 19th century from a protein in cheese. The name tyrosine is in fact actually derived from the Greek word for cheese. Still, I find it convenient to think of tyrosine as hydroxyphenylalanine. Let's keep going. We are going to build 4 more significant chemical compounds. We will make the first compound by modifying the number 3 or orthocarbon of the tyrosine phenyl group indicated in green. In particular, we will remove the hydrogen from this carbon and again replace it with a hydroxy group. This resulting compound is logically called dihydroxyphenylalanine and is usually abbreviated as DOPA, D-O-P-A. Pay attention to the dihydroxy substituted phenyl group. This functional group is known as a catechol. DOPA is both an amino acid and a catechol derivative. The naturally occurring compound dopamine is made by removing the carboxyl group from DOPA. It's important to notice that unlike DOPA, dopamine is therefore not an amino acid. Dopamine is classified as a biogenic amine. Biogenic amines are a group of basic nitrogenous compounds that are often formed by decarboxylation of an amino acid. Like epinephrine and norepinephrine, dopamine is also classified as a catecholamine. Naturally occurring dopamine may be converted to norepinephrine. The enzyme that adds a hydroxy group to the beta carbon of dopamine to create norepinephrine is named dopamine beta hydroxylase. Recall from the previous video lesson that the prefix nor signifies the absence of a methyl group. Therefore, the chemical structure of norepinephrine may be thought of as epinephrine minus a methyl group. Let's look at how naturally occurring norepinephrine is converted to epinephrine by considering the norepinephrine nitrogen atom. If we replace a hydrogen atom from this nitrogen atom with a methyl group, we have epinephrine. As I have alluded, the biosynthesis of these compounds in humans is catalyzed by various enzymes. The key enzymes are named tyrosine hydroxylase, dopa decarboxylase, dopamine beta hydroxylase, and phenylethanolamine n-methyl transferase. This wraps up the second video in this series and our rather brief introduction to the chemical nature of drugs. In the following videos, we will continue our study of pharmacology by examining how drugs affect the cardiovascular system. Please leave questions, critiques, and comments below. We will conclude this lesson with a few figures for your review. Thank you.